Hello, everyone. Welcome to this interactive session for the course Classics in Neuroscience. Am I audible? Yes, sir. And are you able to see my screen? Okay. Let us wait for 2 more minutes, and so that in case others are joining, and then we'll start the session. So, welcome to today's interactive session for the course Classics in Neuroscience. I am Ramya Shekhar, a BMRF TA for the course. So, in today's session, I'll basically I have giving you a set of questions. Just a minute. Yeah, so in today's session, we'll basically be discussing certain questions with regard to whatever you have studied so far. And I hope this is an interactive session where like whoever knows the answer can either unmute yourself and answer, or you can drop your message in the chat box as to which is the correct answer. And there are some questions for which multiple answers might be correct. In those cases, I have mentioned as MSQ, so where there multiple answers are correct. So feel free to unmute yourself and answer, or drop your answers in the chat box. So let's begin with the first question. As you might understand, many of these questions are dispersed throughout like different weeks. So these questions are not with regard to any particular week of the course, but rather it is dispersed throughout the whatever you have studied so far. So, first question: when and by whom was the neuroscience program formed? So, does anyone know the correct answer to this question? Can anyone share your answer in the chat box? Option D. Yes, that is correct. So, Francis Schmidt was the first person who came up with the neuroscience program, and he did so in 1960. Next question: who coined the term neuron? So the options are Charles Sherrington, Wilhelm von Waldeyer, Camillo Golgi, and Santiago Ramon y Cajal. So, does anyone know who coined the term neuron? Yes. So, yes, Voltaire was the one who coined the term neuron. So, although it was Santiago Ramon y Cajal who had proposed that there are independent, so he was the one who had said that there are independent entities or cells in the nervous system, and they they interact with each other, or they communicate with each other through contacts. So although it was Ramoni Cajal who had come up with this idea, the actual term was coined by Wilhelm von Waldeyer. And Cajal had been quite upset because when Wilhelm von Waldeyer had coined this term neuron, a lot of the world actually had thought that it was Waldeyer who had come up with the concept of neuron as well, which was not true, because Santiago Ramon y Cajal was the one who had actually come up with the concept of neurons. But the actual term was coined by Wilhelm von Waldeyer. And Waldeyer had also coined other terms. For example, he was the one who had also coined chromosomes, for instance. So Voltaire actually had like synthesized all of the studies of Golgi, Cajal, and others. So whatever people like were had proposed at that time, all of that Voltaire had synthesized, I think, into a six-part series or something. And during that time, he had also coined the term neuron. So also basically what you need to remember is that although Cajal had come up with the concept or the neuron doctrine itself, the term was not coined by Cajal. The term was coined by Wilhelm von Waldeyer, a German scientist, if I'm not wrong. Next question. Who is regarded as the father of neuroembryology? Is it Paul Weiss, Wilhelm His, William Scoville, or Carl Lashley? Does anyone know the answer? Father of neuroembryology. Yes, Srija. William His is the correct option. So if you go back to the one of the early video lectures in the course, you will see that it was discussed about how like first the studies of Cajal had been were discussed, and then the professor had discussed about Wilhelm His. So Wilhelm His was the one who first thought of this idea of using embryos and fetus, and using embryos and fetus of different ages, and trying to study how the nervous system is developing in these embryos. So that's why he is regarded as the father of neuroembryology. Because before that, people had not actually thought of using embryos to carry out such studies. But Wilhelm His was the one who pioneered such work. So he had used embryos of different ages and tried to compare and study how the nervous system is developing in these embryos. So he had used human embryos, if I'm not wrong. And basically, because at that time they couldn't, I mean, initially the scientists were not actually doing experiments as such. They were only actually observing the development or observing the phenomena. So Wilhelm His was also carrying out observations, but he had decided to use embryos for that. And it was a very good idea, because in embryos, the nervous system, you can actually see how the nervous system is developing. So it is the less complex version of the adult embryo, I mean adult nervous system, right? So it was more easy to observe and understand how the nervous system was developing. So that's why William His is regarded as the father of neuroembryology. So, who was Paul Weiss? Paul Weiss was also a neuroembryologist. He was so. If, we have also come across Paul Weiss in many cases. So, please go back and check what were the contributions of Paul Weiss. William Scoville, if anyone remembers, was the surgeon or the surgeon who had carried out the temporal lobe, the removal of bilateral removal of the temporal lobes in the patient HM. So again, go back and check what William Scoville had done. What was like? How did that help us in our understanding of how memory is formed? So Carl Lashley again was another scientist who also had made certain very important contributions. Again, we have come across Carl Lashley in very many contexts across our discourse. Again, I would urge you to go back and check what he did. Next question. Who coined the term synapse? Was it Santiago Ramon y Cajal, Charles Sherrington, Michael Foster, or Wilhelm von Waldeyer? Okay, what did William Scoville do? So, William Scoville was a surgeon who had, yes, Anushka, the correct answer to the question is Charles Sherrington. Let me just explain what William Scoville had done and then I'll get back to this question. Yeah, so to answer your question, Srija, William Scoville was basically a surgeon who was actually carrying out, like, he was actually practicing other kinds of surgery when he was presented with a patient who had severe epilepsy. So, a lot of seizures, major seizures that person was experiencing. So, that person, the name, we call that person patient HM. So this patient HM, when he was a young child, he had fallen off his bicycle. And ever since then, I think he got hurt from that incident, or maybe he hurt his head. So ever since then, he started having seizures. So initially, his seizures were like minor seizures, but with increasing age, he started having more and more seizures. And finally he started having the grand mal seizures, or the ones that were like major seizures. So he basically had epilepsy, and so much so that he couldn't carry out his normal functions. He started having them like 2 times a day or something. So he couldn't carry out his job properly, and it was getting affecting his daily life basically. So at that time, the only available treatment were barbiturates. But barbiturates used to cause lot of drowsiness. So they were antipsychotic drugs and they used to cause a lot of drowsiness. And now because of that, I mean, it was not possible for him to carry on with his daily life easily for patient HM. So then patient HM was brought to William Scoville, who was a surgeon, like I said. So Scoville decided that let's remove his temporal lobe. So Scoville had not actually performed this kind of surgery before uh, on any other patient, but he decided, okay, let me basically remove both of his temporal lobes. So on both sides, the temporal lobe that is there, he said, let me remove that. So he got rid of both of the temporal lobes of patient HM. And after the surgery, it seemed like patient HM had recovered. But what actually had happened was that patient HM could not form any new memories. So patient HM could not remember anything new that you presented in front of him. If a person came in front of him, that HM would not remember him after some time. He would never remember encountering that person. If you made him do some simple, like if you made him try to remember some simple numbers or some simple words or calculations, he would forget them after a while. He also had so this was called anterograde amnesia. So the ability to form new memories was lost. And he also had about 3 years worth of retrograde amnesia. So his previous memories, some of them were also lost. So then what happened was William Scoville shared this finding that when the temporal lobes were basically removed, patient HM had experienced so and so, and he saw that these memories were not being made, and all of this. And then William Scoville basically brought into the picture Brenda Milner. So Brenda Milner was a psychologist. So Brenda Milner and William Scoville together studied patient HM and tried to find out why patient HM is not able to form memories and to what extent is he able to form memories. So basically, they tried to understand what had happened to patient HM and how the surgery performed by Scoville had basically impacted the memory formation abilities of patient HM. So they had found out what is the role of the hippocampus in that and what had actually happened to patient HM and so on. So it basically gave us a lot of information about the different. As far as I know, HM did not have any difficulty in language comprehension, Ananya, but I might have to go back and check. But as far as I remember, I don't think he had any problem with that. What I do remember is that even after they performed this surgery, patient, like initially they thought that initially they thought that this seizures problem is gone. That's what they thought. But even after the removal of the temporal lobes, after some time, his seizures actually returned. So there was no use in carrying out this surgery in terms of treating his epilepsy. But patient HM also ended up having no ability to form new memories. So then they try to understand that what is so initially they had thought that there is some atrophy in the hippocampus and so on, and later they try to understand what is happening with patient HM and why was he not able to form memory. So they carried out a lot of studies on patient HM. So he became very even without his knowledge, patient HM became kind of the most famous patient in the history of neuroscience, you can say. Yeah, so that is all about who William Scoville was and what he did and his role in basically neuroscience, like in our understanding of memory and so on. Of course, there were a lot of people who later questioned the practice, the ethics of the surgery that Scoville had carried out on HM. Like, why did William Scoville have to remove both of the temporal lobes? It was something he had not done before, and it was something he did not also have expertise on. So, why did Scoville carry out this kind of an experiment? And if you see, according to today's standards, of course, that kind of experiments or that kind of surgeries would not have been permitted. But during those times, there was no regulation. Like, if you are a surgeon, if you think that something should be done, that's it. Good, it is done. So yeah, these are the kind of things that happened at that time. Of course, they led to a lot of increased understanding in many different fields, but they also had their problems. Okay, I hope that is clear to you, Shrija. Coming back to the question, who coined the term synapse? So, as correctly answered by Anushka, as correctly answered by Anushka, the correct answer was Charles Sherrington. So, uh, initially, I think this happened Charles Sherrington coined this term, I think in the year 1897, if I'm not wrong. So, Sherrington agreed with Cahal's proposal that there are indeed these gaps between neurons, and that neurons basically are not a reticular network the way Golgi was proposing. Rather, they are separate independent entities which are interacting with each other. And these regions where the neurons were interacting with each other, these regions or these gaps, that those were called by Charles Sherrington as synapse. And there's also a story behind this. So So how did this happen? So remember Michael Foster? So Michael Foster was actually like who wrote, he was someone who wrote a textbook on physiology. So for his textbook on physiology, Michael Foster, so that book was very famous. So the textbook of physiology that Michael Foster had published was actually very famous. So Foster wanted Sherrington to contribute some some chapters on his related to what Sherrington was doing to Foster's book. So at that time, Sherrington had come up with, like in what Sherrington had written, he had proposed the term synapse for these uh, connections or these gaps that exist between neurons. And actually, Sherrington had consulted with a friend of his. So he had consulted with Veral, I think, that what term he should come up with. And they had had a discussion with each other. And Veral had suggested that why don't you call the term as synapse? So because synapses means basically connection or contact. So he said, why don't you call the term as synapse? So using that, Sherrington coined the term synapse for what we now know as the connection or the gap between two individual neurons where they communicate with each other. I hope that is clear to you guys. Okay. Moving on to the next question. What stain did René Cotiu initially use for studying the neuromuscular junction? Is it the black reaction, the Coel method, the Janus green B stain, or the Nissel stain? Who knows the answer to this question? Any volunteers? Anyone knows the answer? No, Sheja, it's not the black reaction. The black reaction is not the correct answer. Anyone else knows? Or any other answer? So the correct answer is Green B. So Janus Green B is a dye that René Cotiu had used to study the neuromuscular junction. So this he had stained with this dye to show that there is indeed a neuromuscular junction that exists and what is the exact structure of the neuromuscular junction and the fact that acetylcholine is released from the presynaptic site and goes and interacts with the postsynaptic the basically the muscle that is present on the postsynapse. So this was the dye that René Cotiu, who was a French scientist. So René Cotiu was a French scientist who was working in Paris. So he had he started studying the neuromuscular junction, and one of the first dyes that he used was this Janus Green B to understand what is exactly the how it I mean what is the how it works and what are the structures and so on. And later, after George Coel had developed the Coel method, at that time, Cotiu also modified the Coel method and used that for his later stainings and experiments with the neuromuscular junction. But initially, he used the Janus Green B stain. So he had made a very important contribution to the neuromuscular junction. The fact that acetylcholine is released from the presynaptic neuron and goes and binds to its receptors on the postsynaptic muscle, and that there is an acetylcholine esterase that is present in the synaptic cleft, and so on. So the correct answer to that question is Janus Green B dye. So the Janus Green B stain was the one that René Cotiu had used. Next question. Which discovery is attributed to Betty Guerin? Does anyone know? So, is it the discovery of the structure of the synapse? Is it the discovery of the Renshaw cell? Is it the discovery of the nodes of Ranvier? Or is it the discovery of the structure of myelin sheath? Yes, Ananya, the correct option is option D. You are right. It is the discovery of the structure of myelin sheath. So Betty Guerin had shown that it is, so how is myelin sheath formed in the peripheral nervous system? So she showed that basically the Schwann cell, the Schwann forms an envelope around the axon, and basically that is the struct. I mean this envelope that is formed, the continuous looping of this the Schwann cell around the axon is what actually forms the myelin sheath. And that was an extremely important discovery because it later improved our understanding of how the how the there is saltatory conduction, right? So basically, because of the presence of myelin sheath, what happens is that myelin sheath acts as an insulation. So basically, only at the nodes of Ranvier, there is basically faster passage of the neuronal impulse, because only at the nodes of Ranvier the information can get transmitted, because the rest of the axon is basically covered by the myelin. So the myelin is a fatty substance that insulates the axon, and only at the nodes, or the basically the nodes of Ranvier, is where there is transfer of the neuronal impulse. So the information travels much faster because of myelination. And therefore, this discovery was very important because it improved, I mean, basically gave us an understanding of what is the importance of glial cells, especially, and what is the importance of myelin and how myelin works. And all of this came because of the work that was done by Betty Guerin in the peripheral nervous system. So, yes, the correct answer is discovery of the structure of myelin sheath. Next question. Who was the first to use the term electrophysiology? Does anyone know? Is it Luigi Galvani? Is it Walter Nurst? Is it Carlo Matteucci? Or is it Alan Hodgkin? No, Anushka, Galvani is not the correct answer. No problem. Why are you saying sorry? Remember, we are all trying to learn over here. There is no need to say sorry. As long as we learn and we understand the concepts, that's all that matters. So the correct option is Carlo Matteucci. So Luigi Galvani had only shown that there is bioelectricity. So he had used frogs to study, like if you the fact that there is electricity in the nerves, and that is what is responsible for muscle contraction. So he had basically come up with, I mean, he was the first one to show that there is bioelectricity, or the fact that nerves conduct electricity, and that is responsible for muscle contraction. That is what Luigi Galvani had shown. Nernst was the one who came up with the Nernst equation, as we know, which we often study in chemistry and also in, for example, in say biophysics and so on, right? So that was like the Nernst equation was basically was because of Walter Nernst. Alan Hodgkin was the one who came up with the Hodgkin-Huxley model for how the action potential basically travels. I mean, how is the action potential initiated and how nerve impulses actually propagate along the axon. So that was proposed by Alan Hodgkin and Huxley. So it was Carlo Matteucci, who again belonged to the same nationality as Luigi Galvani, who came up with the term electrophysiology. So the correct answer is Carlo Matteucci. Next question. Which theory is attributed to Julius Bernstein? Is it the contact guidance theory? Is it the membrane theory? Is it the resonance theory? Or is it the cell theory? Yes, both Anushka and Shrija, you are correct. It is the membrane theory that is the correct answer. So Julius Bernstein had come up with the membrane theory to explain how the fact that there is a membrane potential, there is a resting membrane potential, and that the during a neuronal impulse, maybe there is a change or a disruption in this resting membrane potential. So Julius Bernstein and was one of the individuals, and there was also Overton. So the membrane theory is also called as the Bernstein-Overton theory. So Overton had also proposed some aspects of the membrane theory, and Bernstein had proposed certain aspects of the membrane theory. So we know that the contact guidance theory was proposed by Paul Weiss, remember? The neuroembryologist, the one whom I had discussed in one of the previous contexts. So Paul Weiss was actually the person who proposed the contact guidance theory. And the resonance theory was also proposed by Paul Weiss. And the cell theory was proposed, like we know, by Schladen and Schwann, and also had inputs from Virchow. So again, go back and check what the contact guidance theory and the resonance theory are. Those were also some things that were discussed uh, in the course when the for the both the contact guidance theory and the resonance theory relate to how the neuron find its target, basically. So how the neuron knows that which target it's supposed to contact with. So both the contact guidance theory and the resonance theory were proposed in that context. And of course, the cell theory was basically that all cells, that all organisms are composed of cells, plants also are composed of cells, animals are also composed of cells. And Virchow said that cells exist from pre-existing cells, omnis cellula acellular. So basically, that is the cell theory. And like I said, membrane theory is to do with the membrane potential, that neurons have a certain membrane potential, the resting membrane, which is called the resting membrane potential, and so on. So the theory that is attributed to Julius Bernstein is the membrane theory. Next question. Who discovered the axonal growth cone? Was it John Eccles? Was it Henry Dale? Was it Santiago Ramon y Cajal? Or was it Charles Sherrington? Yes, Shreja, you are right. It was Cajal who discovered the axonal growth cone. He was the one who actually proposed the term axonal growth cone as well. So when Cajal was, so you know that, I mean, we know that Cajal was the one who came up with the neuron doctrine, right? So when Cajal was obsessed with basically looking at all kinds of neurons and different parts of the nervous system under his microscope. So, a little background about Cajal. Cajal was a Spanish scientist who basically came from a small village in Spain, and initially he wanted to be an artist, but his father like strongly opposed that, and his father decided that he should do basically practice medicine. So on his father's insistence, Cajal basically studied medicine, and he started, I mean, he took up a position after failing two to three times. He finally got a position at a university. And at that time, he started he basically got interested in looking at different parts of the nervous system under the microscope. So he during his time in the military, he had whatever money he had saved, he put all of that into buying a microscope, and using that microscope he used to observe the different sections and different parts of the nervous system all the time from different animals. So while he was, and he used to make very elaborate and very intricate drawings of whatever he used to see under the microscope. So, like I said, he was a very good artist, and he used to spend all of his time, weekday, weekends, days, and nights in the lab, just trying to look at the neuronal cells and make different drawings. So he had made drawings of the pyramidal cells. He had made like lots of drawings of different types of cells in different parts of the nervous system. And while he was doing that, he observed, and he made this comment that the axon of the neuron probably has a tip, which is a specialized structure, which he called the axonal growth cone. So he called that the tip of the axon. He said that this tip of the axon, basically the it is a specialized structure whose role is to basically navigate through all the crowd and all the traffic that is there in the nervous system, like a with a single minded focus, that the tip basically navigates across all of these across this traffic and this crowd, so that it can reach its specific target. So he compared it to a battering ram, which will not stop at anything, which will just push through so that it can reach its target. So that is how Santiago Ramon y Cajal proposed the axonal growth cone. And now we do know that there is actually a structure like that. So when axons are forming new contacts, they do actually have this growth cone, which is a specialized structure, which is rich in cytoskeleton elements like actin, so that the growth cone can establish contact with the target cell. So the one who discovered the axonal growth cone was Santiago Ramon y Cajal. and Henry Dale, John Eccles, Charles Sherrington. These were also scientists. They were also neuroscientists. We had spoken about Charles Sherrington, right? So Charles Sherrington was actually Sir Charles Sherrington. So he, like I said, was the one who had coined the term synapse. So Charles Sherrington had coined the term synapse. Charles Sherrington was working on the he worked extensively on the spinal cord. So Charles Sherrington basically contributed to our understanding of the reflex action, so the reflex arc, and the fact. So he was the one who said that when there is when one muscle is contracting, its antagonist muscle is relaxing. So that is one of the things that Charles Sherrington had proposed. And John Eccles was basically thus one of the students of Charles Sherrington, in fact his last student. And Charles Sherrington was basically, like you many of you might know, was from England. So Charles Sherrington was from England. John Eccles was an Australian who he was a son of school teachers, and he had after doing his preliminary studies in Australia, he had gone to England to work under Charles Sherrington. So John Eccles was an Australian who also carried on Charles Sherrington's work on the spinal cord, on understanding reflex action. John Eccles found out the whole synaptic pathway for the reflex action. He made many other important contributions. Again, go back and check what were the contributions of John Eccles and what were the awards or the prizes that John Eccles received. Again, Henry Dale was another important pioneering scientist who made the observation that there are chemical substances that are released at the synapse. So he was one of the first people who proposed that there are chemical substances like acetylcholine which are released at the synapse. So go back and check all of that. Also check about the debates that used to happen between John Eccles and Henry Dale. So I know I'm deviating from the question. We have already discussed that the answer is Santiago Ramon y Cajal, but you should also know about these other scientists and their contributions. That those are also important for your understanding of the course. So do go back and also check the debates or the heated arguments that used to happen between Eccles and Henry Dale. And also go back and check what Dale's law was. So I'm trying to help you recap all of these things for your upcoming exam. So I hope that is clear. I hope no one, like if you have any questions, feel free to drop them in the chat box or unmute yourself. Either way is fine. Okay, Anushka. So, next question. In which year, in which year did Watson and Crick publish their model of the DNA double helix? So, who knows the right answer? So, is it 1947, 1966, 1953, or 1943? 1953. That is absolutely correct. Both Shija and Ananya, you are correct. 1953 is the correct answer. So, who knows how this happened? Who knows the story about this? How they came up with the model, how they published this, where they published it, and so on. Who can tell me the story? Even if you're like not able to completely say the answer, that's also fine. I just want to help you understand what happened. That's it. Anyone knows? Yes, they used X-ray diffraction data, but was that diffraction data their own data? Was it the one, had they carried out any experiments to come up with that X-ray diffraction data? Yes, they had used Rosalind Franklin's X-ray diffraction data. So Watson and Crick had not performed any experiments of their own. They were on. So at that time, there was this race to find out the correct model for the structure of the DNA. So everyone wanted to be the first to come up with a model. So even Niels Bohr had published a model. So Niels Bohr, who you must have come across in physics and chemistry, right? So even Bohr had actually come up with a model of the DNA, but that was actually not a correct model. So at that time, Watson and Crick. So Watson was actually an American who had come to England to carry out, I think, postdoctoral work, if I'm not wrong. And Crick was already in England, and he was also trying to find out something interesting to do. So Watson and Crick immediately gelled well with each other, and they used to have the lots of discussions. They used to try to come up with what might be an actual model of the DNA, but they were actually not carrying out any experiments of their own. So then at that time, they interacted with Morris Wilkins. So during their interaction with Morris Wilkins, so Wilkins actually showed them some of the X-ray diffraction data that his colleague Rosalind Franklin had been carrying out. So, but Rosalind Franklin did not know that Watson and Crick had been taking a look at her data. So she was not aware of that. But Morris Wilkins had shown her data, her X-ray diffraction data on DNA to Watson and Crick. So Franklin had been doing some experiments with her student, and Wilkins was her colleague. So he showed her data to Watson and Crick. And when they saw that, they were immediately able to understand. So when they took a look at that data, the X-ray diffraction data, they were immediately able to understand what the structure of the DNA might be. And then they came up with a model. They actually built a physical model of DNA as a double helix. So double helix because it has two strands, and the two strands of DNA, they are helical so they are arranged as a helix, and they are the two strands of DNA run in an anti-parallel direction to each other. So the one runs from a 5 prime to 3 prime direction, the other runs from a 3 prime to, so one runs from 5 prime to 3 prime, the other one runs from 3 prime to 5 prime. So they are anti-parallel in nature. So they came up with this model of the DNA double helix. And because of that model, they published this work in Nature in 1953. And in that same issue of Nature, Rosalind Franklin and Morris Wilkins had also published their work in as separate paper. So in the same issue, these two had published in Nature. One Watson and Crick obviously published their model of the DNA double helix, and Rosalind Franklin and her Wilkins had also published the X-ray crystallography data that they had come up with. But the world acknowledged Watson and Crick and Wilkins, in Watson and Crick much more actually, and before they got the Nobel, when the Nobel Prize was awarded, Franklin actually died a little while before the Nobel Prize. So she was not awarded the Nobel Prize because she had died. And Nobel Prize is not awarded after one's death. So only Watson, Crick, and Wilkins received the Nobel Prize for their work towards the understand our like elucidating the structure of DNA. And like Franklin, basically, you can say was her contribution was kind of, you can say, ignored or not appreciated. And like many other women scientists, so like Rosalind Franklin became, and you can say, an example or a model for how women scientists actually have to struggle much more than their male peers to be recognized in academia for their work, and how it is much more difficult for them to be appreciated for their work, to carry out research in academia, and the kind of discrimination they face as women scientists. Yes, Anushka, Morris Wilkin had helped Watson and Crick, like you correctly pointed out, and he also had a share in the Nobel Prize. So Watson, Crick, and Wilkins, all three of them received the Nobel Prize. Next question: who introduced the term dendrites? So was it Wilhelm His? Was it Wilhelm von Waldeyer? Was it John Eccles? Or was it Roger Sperry? So who introduced the term dendrites? It's okay even if you're wrong, it is fine. As long as you volunteer, as long as you try to answer. And remember, when you try to answer and suppose you are wrong, even then, like if you are corrected and if you understand what the right answer is, you will always remember it better because you actually made an effort to understand the question and the concept. So it was always important to try to understand better. Even if you stumble, even if you fumble, even if you're wrong, it's all right. After all, we are all trying to learn. That's what matters, right? In the end. Yes, Ananya, you are right. Just a second. Yes. So who introduced the term dendrites? The correct answer is Wilhelm His. So William His, uh, so at the time these were called as protoplasmic protrusions. So William His was the one who said, let us call them as dendrites. And when he introduced this term, others quickly started adopting the term dendrites. So William His, the famous neuroembryologist or the father of neuroembryology or the founder of neuroembryology, was the one who introduced the term dendrites. We have already discussed these other scientists, right? We have discussed the who Wilhelm von Waldeyer was. So Wilhelm von Waldeyer was a German scientist who was very good at coming up with terms or names. So he Wilhelm von Waldeyer had come up with the terms for neuron. He had come up with chromosomes and so on. But he was not the one who came up with dendrites. Dendrites was actually coined by Wilhelm His. John Eccles, we discussed that John Eccles was a Nobel laureate who had worked on understanding different aspects. So he had worked on central synapses. So I'm talking about John Eccles, okay? So John Eccles had worked on central synapses. He had worked on understanding the inhibitory postsynaptic potentials. He had elucidated the synaptic pathway in the spinal cord. So John Nichols had actually done a lot of different kinds of work, but again, he was not the one who introduced the term dendrites. Roger Sperry was again another Nobel laureate who basically worked on understanding. He proposed the chemo affinity hypothesis. I will come to that in some time. So there are some other questions where we will be dealing with that. So I will explain to you the work of Roger Sperry. Why did he win the Nobel Prize? What did he win the Nobel Prize for? And so on. Okay, so the correct answer to this question is Wilhelm His. Is that clear? Okay. Next question. Which of the following scientists were proponents of the neuron doctrine? So this is a multiple select question. There are multiple correct answers. So who knows which are the correct answers to this question? So the options are Luigi Galvani, Santiago Ramon y Cajal, Camilo Golgi, and Charles Sherrington. So, which scientists among these were the proponents of, or the ones who supported the neuron doctrine? So, when you say proponents, you mean that they supported the neuron doctrine or they advocated for the neuron doctrine. So, they were the ones who said that neuron doctrine is correct. So, which of these scientists were proponents of the neuron doctrine? So, there are multiple correct answers to this question. Anyone? Yes, it was Cajal and Sherrington. So the correct answers are Santiago Ramon y Cajal and Sir Charles Sherrington. So again, Luigi Galvani was the one who proposed that there is bioelectricity. So at that time, it was too early. It was the neuron doctrine was not even in the picture. Camillo Golgi said that the nervous system is composed of nerve nets, or basically he said that it is he was a proponent of the reticular theory, or he said that the nervous system is basically a reticular network. It is all interconnected. All the neurons, I mean, all the components of the nervous system are basically interconnected in terms of a net. There are no separate entities. They're all connected to each other. The function of the cell body is to just provide nutrients to these nerve nets, and it's all a reticulum or an interconnected network. That is what Camillo Golgi said. So he was completely opposed to the neuron doctrine. He was and he was an he did not agree at all with the neuron doctrine. Instead, he agreed with the reticular theory. Santiago Ramon y Cajal was the one who actually proposed the neuron doctrine. So Ramon y Cajal said that the nervous system is composed of independent neurons or independent entities or cells which probably interact with each other or they communicate with each other. They are independent entities, but they communicate with each other. That is what he said. He said that they are in touch with each other, but they do not touch each other. So they do not actually physically form a network as such, the way Golgi was proposing. Rather, there are sites of contact between these independent entities. So that is what Cajal said, and that is what came to be called as the neuron doctrine. And like I said, was Charles Sherrington agreed with Cajal, and he also said that the in the cells of the nervous system are independent, like Cajal was proposing, and he was the one who said that there are these points of contact or these gaps which exist, and he said that let's call them as synapse. So both Cajal and Sherrington were proponents of the neuron doctrine, whereas Golgi was opposed to the neuron doctrine. And do you know, if you go back and read the Nobel, so both Santiago Ramon y Cajal and Camillo Golgi won the Nobel Prize. So even Charles Sherrington won the Nobel Prize for that matter. But Cajal and Golgi won the Nobel Prize together. In the same year, they both won the Nobel Prize together. So first, Golgi went on the podium and he gave his talk. So Nobel laureates give a Nobel lecture. So they give a talk. So Camillo Golgi first went to the stage and he gave his talk in which he completely spoke in favor of his reticular theory. Even at that time, he was not willing to accept the neuron doctrine that Cajal had proposed. So he completely as in he when he gave went onto the stage and gave his lecture, he he was completely against the neuron theory, even on the same stage as Kahal, and he favored his reticular theory. Then, after Golgi had finished his lecture, next Ramoni Kahal went on the stage to give his Nobel lecture, and he spoke about his evidences which showed that the neuron doctrine was correct. So they kind of, you can say, had a rivalry between them. Although, interestingly, Kahal, like to study all of his preparations of the nervous system under the microscope, Kahal actually used the stain that was developed by Golgi. So, if you remember, in one of my previous questions, we had there was this option, right? The black reaction, correct? So, the black reaction was actually developed by Camillo Golgi. So Camillo Golgi had developed had developed the black reaction, or basically it was a staining method that Golgi had developed to stain the neurons. But the problem is that this staining method that Golgi had developed, it was not very reliable. So sometimes it used to work, sometimes it did not work well. Sometimes the structures used to stain properly, sometimes they were barely visible. So Golgi was also a practitioner of medicine. He was also basically a doctor. So when he was working in a small hospital, a rural a kind of not well-to-do hospital, small hospital. At that time, Golgi, in the kitchen of that hospital, he had set up this small laboratory, and in that laboratory, he had come up with this staining technique, which is called the black reaction. Basically, that staining technique basically resulted in the deposition of silver, so that silver precipitate used to make the uh, used to stain as a black precipitate, so that's why it was called the black reaction. But when so that was developed by Golgi. But later, like I said, the this stain, this Golgi stain or the black reaction was not very reliable. But later, so much after Golgi had developed this stain, Kahal got to know about this team. So Kahal had visited one of his acquaintances, and that acquaintance had gone to several places and all, and he came back with a lot of information and specimen and so on. So when Kahal met with this acquaintance to discuss about what is new and so on, so he got to know about this black reaction, this Golgi stain that Camillo Golgi had developed. So Golgi was an Italian. Cajal was a Spanish scientist. So Cajal got to know about this stain, and he was amazed. He was completely blown away by this staining technique. And he decided that he will improve this staining technique and he will use it to study the nervous system. So in around, I think, 1887 or so something, he said that I will start. So initially he was working on many other things also, not just the nervous system. He was also trying to study other things. But in 1887, he said, I'm going to drop everything else. I'm going to focus only on the nervous system. And I'm going to improve the staining technique and I'm going to use it. He spent, I think, around 6 months or something just trying to improve the staining technique. So that one staining technique, he tried to use different concentrations, different incubation times, different sections of tissue, and different doing the same staining 2 times. So different kinds of ways he employed to improve the staining technique that Golgi had developed. And after 6 months, he finally perfected the staining technique, and then he used it for all sorts of nervous tissue from different animals, from different parts of the brain, and so on. And he used that and he made all of his drawings and sketches and all of that, and he came up with the neuron doctrine. So Kahal was very Kahal had called Golgi as the savant of, so from he regarded Golgi actually as, he regarded Golgi a lot. But Golgi, because Golgi, the way Golgi spoke during his Nobel lecture, Kahal was very hurt by that. So Kahal was like, because Golgi still would not accept the neuron doctrine, and the way Golgi spoke in his Nobel lecture, Kahal was quite disappointed with that. But Kahal actually used the technique that Golgi had developed for his work, and he came up with the neuron doctrine, which was completely opposed to that of what Golgi was proposing. And now we know that it was the neuron doctrine is actually the correct hypothesis, or the correct. I mean, neuron doctrine was correct. What Golgi had proposed was actually not correct. The reticular theory was incorrect. The neuron doctrine was actually correct. So there are individual neuronal cells that exist, and they uh, communicate with each other. They are not a network. So that is the story of Camilo Golgi and Santiago Ramon y Cajal. And interestingly, if you go back to some of the previous lecture videos, you might also, where the professor talks about Cajal's background and so on. Cajal actually was even offered the position of education minister in Spain. But Cajal declined. He said that no, I want to focus only on academia, on my research. I don't want to do. I don't want to become a minister and all. He helped in like coming up with certain reforms and rules and so on. But he said no, I don't want to become an education minister. I will just focus on my research. So there is a Cajal Institute that was constructed just to honor Cajal's memory. I think the Cajal Institute is in Spain, Madrid, if I'm not wrong. So it was while Cajal was alive, this Cajal Institute was constructed in to honor Cajal's work and his contributions. So that is a little history about Cajal and Golgi. Do go and take a look at their Nobel laureate lectures. So the each Nobel laureate has Nobel lectures. If you go to the Nobel Prize site, you will find the original lectures that they had proposed, and what they had, like, what was, you basically will give you a good idea about what they thought or how they came up with an idea like that. So I hope that is clear. Okay. Next question. Which of the following, sorry, no, sorry, said this. Which of the following were the contributions of Bernard Katz? Again, it's a multiple select question. So multiple answers are correct. So the options are the GHK equation, the ultrastructural identification of synaptic vesicles, quantal release of neurotransmitters at the neuromuscular junction, discovery of the nerve growth factor. So, which of the following were contributions of Bernard Katz? B and C. You are not completely correct, Anushka. One of the options is correct, one of the options is not correct. So now, and also there is another option which is correct. Any other guesses? Of course, C is correct. But any other option which is correct other than C? I already said C is correct. So let's mark C as correct. Any other option? So the other options that are remaining are discovery of the nerve growth factor, ultrastructural identification. So ultrastructural identification of synaptic vesicles is wrong. So this is wrong. No, Anushka, NGF is not correct. This is not correct. So the GHK equation is correct. Sorry. So the GHK equation and quantal release of neurotransmitters at the neuromuscular junction. So let me explain. GHK equation is basically the Goldman Hodgkin Katz equation. So GHK stands for Goldman Hodgkin Katz equation. And yeah, so option A and option C are correct. So why? So these three individuals, these three scientists, Goldman, Hodgkin, and Bernard Katz together came up with this equation to derive the different concentrations of different ionic concentrations of uh, like say of sodium and other ions at the which are responsible for basically the membrane potential, the resting membrane potential of the um, uh, membrane of the nerve cell. So that was the Goldman-Hodgkin-Katz equation. Ultrastructural identification of synaptic vesicles was actually, so when I say ultrastructure, I mean by electron microscopy. So ultrastructure means the images that you take by using electron microscopy. So who did the ultrastructural identification of synaptic vesicles? So there were different scientists. There was Pallard and Paley, and there was D. Robertus. So these were the scientists who actually initially like took electron microscopy images of the synaptic vesicle of the synapse and observed the presence of synaptic vesicles. So those scientists, so like I said, Pallard, Paley, and D. Robertus. So these scientists were the ones who actually first like it is debated that which among them was the first to do it, which among Pallard and Paley and D. Robertus was the first to do it. But these were the among the first scientists to actually look at the synapse and observe these synaptic vesicles. So that was not done by Katz. But what Katz did was that he observed, he made recordings, intracellular recordings at the neuromuscular junction. So he, together with other scientists like Paul Fatt and Del Castillo, so, together with those people, Bernard Katz actually carried out intracellular recordings at the neuromuscular junction. And by carrying out these intracellular recordings, he observed that there are these miniature end plate potentials or MEPPs. So he saw that there are these miniature end plate potentials at the, so if you record from the muscle, you are able to see that there are these very small amplitude and potentials that you can record even in the absence of any nerve stimulation. So you're not stimulating the nerve, but still you're able to see these miniature or very small end plate potentials at the neuromuscular junction, and these were very spontaneously released. There was no pattern or something. They were just spontaneous. These end plate potentials were all spontaneous, random, and there was no stimulus, but there was no stimulation of the nerve, but still he was able to record those. And by observing these miniature end plate potentials, Katz actually came up with the idea of quantal release of neurotransmitters. So he said that there must be some way by which the neurotransmitters are probably released as discrete packets or packages. So there are like each time there is a random spontaneous release of a certain packet of neurotransmitters from the presynaptic site or the nerve. And because of this release of this discrete packets of neurotransmitters, that is why he was able to detect those spontaneous miniature inlet potentials or very small values of uh, potential at the postsynapse. So he said that it's a quantal release, or there's like packages of neurotransmitters. And later, around the same time or a little later, at that time in the 1950s, when the electron microscopy had also started to be like in electron microscopy was also had come into the picture. And like I said, Pallard and Paley, and independently also D. Robertis, these scientists were trying to look at the synapse under the under the electron microscope, and using their ultrastructural image. I mean, using the ultrastructure of that, they were able to observe the presence of these synaptic vesicles. So when Cards came to know that, oh, there are these synaptic vesicles that are there, so he could relate the synaptic vesicles to his idea of quantal release of neurotransmitters. Because it makes sense, right? If there are synaptic vesicles, then a certain number of neurotransmitters can be packaged in each of these synaptic vesicles. And each time one of these synaptic vesicles fuses with the membrane of the presynapse, the neurotransmitters will get released. So each time one synaptic vesicle releases a certain number of neurotransmitters, that is going to be detected at the post synapse. So that is why he was able to see the MEPPs. So each time the synaptic vesicles underwent a fusion and released the neurotransmitters by exocytosis, there was a miniature end plate potential that was observed at the postsynapse. And when there was when the nerve was getting stimulated, if you actually deliberately stimulated the nerve, in that case, when the action potential traveled all the way to the synapse, that caused, so at the time Cards didn't know how it happened, but that caused the release of several synaptic vesicles. I mean, that caused the fusion of several synaptic vesicles to the presynaptic terminal. So it increased the frequency and rate of fusion of synaptic vesicles to the presynaptic terminal, and that caused an evoked end plate potential at the postsynapse. So when it was just spontaneous without any stimulation, that he was able to observe the miniature end plate potentials. And when he stimulated the nerve, that caused the fusion of many synaptic vesicles at a time with the presynaptic membrane, and that caused the evoked end plate potentials. And Cards was able to show that the evoked end plate potentials were basically integer multiples of the miniature end plate potentials. So he was able to show that it is indeed a quantum phenomena, that basically the neurotransmitters are released in discrete packets. That is why the evoked end plate potential was an integer multiple of the miniature end plate potential. So because synaptic vesicles are individual entities, right? So if there are, say, 10 synaptic vesicles or 20 synaptic vesicles, so that is going to really cause some kind of evoked end plate potential, correct? You cannot have half a synaptic vesicle, you can't have 10.5 synaptic vesicles, you can't have 20.2 synaptic vesicles, right? It is also always going to be an integer. So that is why the end plate potential is also always going to be the evoked end plate potential is always going to be an integer multiple of the miniature end plate potential. So that's how Cards came up with the quantal release of neurotransmitters at the NMJ. The idea that there is a quantal release. and he was able to correlate it with the findings of other scientists that there are these synaptic vesicles that are present at the NMJ. So he himself did not identify the synaptic vesicles by electron microscopy, but he was able to use that information to correlate with his idea of quantal release of neurotransmitters. He was not the one to discover the nerve growth factor. He had no role to play in that scenario. That was done by a completely different group of scientists. In one of the future questions, I'll be talking about that. But cards did not have any role to play in the discovery of the nerve growth factor. And by the way, cards received the Nobel Prize for his work on the to show by showing that there is quantal release of neurotransmitters at the NMJ. So he got the Nobel Prize for this work. I hope that is clear to everyone. How does MEPP help with action potential? No, Shrija, MEPP does not help with action potential. What I'm trying to say is that wait, let me explain with a diagram. I think that will be easier for you to understand. Uh, just a minute. Yeah. So, okay. So let us assume we are looking at the neuromuscular junction. Okay. So this is the presynaptic nerve, right? This is the uh, postsynaptic uh, basically is the postsynaptic we're talking about NMJ, so it's going to be the muscle. Okay. So what you mean by MEPP? So MEPP is miniature end plate potential. So what Cards was doing was that, so this is the he was basically using this he was using the frog, and what he was doing was that he was recording from he was doing intracellular recordings from the neuromuscular junction. So when he was recording from the muscle, even when he had not stimulated the neuron, so there was no action potential from the in the neuron. Okay. So when generally what happens, there's an action when you stimulate the neuron, there is an action potential, right? So he had not stimulated this axon of this neuron. So this axon was not stimulated, so there was no action potential in this uh, neuron. So even when there was no action potential in this neuron, he was saying that when he was doing the intracellular recordings, he was saying that when he was recording from this muscle, he was seeing some small, very small amplitude. He was able to changes or fluctuations in the membrane potential. You can see that there is this background noise, and then there is this there's a small fluctuation, right? Again, background noise, suddenly there's a small fluctuation, and so on. And these are all random. You can see that there's no pattern over here, right? There is no like particular time difference between these MEPPs. There is no particular. It's not like there are two of them coming together. Then there is two. Nothing like that. So there was no pattern in this, but he was able to see that there are these very definite small amplitudes. The size is very small. You can see it's like some 0.5 volt difference, right? So he was able to see these very small MEPPs. He called them as miniature end plate potentials. Miniature means small, right? So miniature end plate potentials he was able to see. And they were all very spontaneous. Okay. In contrast, when he was actually he was actually stimulating this, sorry. when he was actually stimulating this nerve, that is, when there was actually an action potential in this nerve, which was propagating along this axon of the nerve, and then it came over here. And by some phenomenon that Katz did not know, he was saying that when there was an action potential in this nerve, he was saying that when he was recording from the post synapse, that is from the muscle, he was able to see this very large peak. Okay, so when there was no action potential in the presynapse, he was seeing only these small peaks, randomly distributed spontaneous peaks. When there was an action potential in the nerve, when he actually gave a shock, when he actually stimulated the nerve, he was able to see this kind of a peak. Okay, so this was called evoked end plate potential, and this was called miniature end plate potential. Okay, so when there was no action potential in the nerve, he was able to record the MEPPs. When there was action potential in the nerve, he was able to record the evoked end plate potentials. Okay. So then what did that mean? Now let us understand in today's context what that means. So in today's context, what that means is basically what he was recording was that. So when there was no action potential that was propagating along this nerve, without any action potential, randomly some of these synaptic vesicles were fusing with the presynaptic membrane. So you can see that these are fusing over here, right? So there is a whole there's a whole machinery over here which we'll not go into right now, but there's a whole machinery over here which involves several proteins and complexes and so on, which help in the fusion of these synaptic vesicles with the presynaptic membrane. So these synaptic vesicles fuse with the presynaptic membrane and release the neurotransmitter into this synaptic cleft or this gap. So randomly, one or two synaptic vesicles used to fuse with the presynaptic membrane and release their neurotransmitter content into the synaptic cleft. And these few neurotransmitters which got released used to go and bind with their cognate receptors on the postsynaptic membrane. Okay, so randomly just one or two used to fuse, and this phenomenon used to happen when there was no action potential in the presynaptic membrane. But when there is an action potential in the presynaptic membrane, what happens is that there is a whole process which happens over here, because of which many of these synaptic vesicles fuse with the presynaptic membrane. Okay, so lot of them fuse at the same time, and there is an increased chance of their fusion with the presynaptic membrane. So at a time, a lot of neurotransmitters get released. And because a lot of these get released, and they all, many of them go and bind with their receptor on the postsynapse, that is why you're able to see such a large, just a minute, such a large dramatic peak, which is the evoked end plate potential, because of a lot of neurotransmitters which are interacting with their receptors on the postsynapse. So MEPPs are not, sorry. So MEPPs are not helping with action potential. MEPPs are those which you can record even in the absence of action potential in the nerve. I hope that answers your question, Sreja. Next question. With which of the following scientists did Paul Fat collaborate? This is again a multiple select question. The options are Charles Sherrington, Bernard Katz, Rita Levi Montalcini, and John Eccles. So, which of them did Paul Fat collaborate with? Anyone wants to answer? So Ananya says B. Yes, B is one of the correct options. We just discussed about in this question. We just discussed about um, the fact that Paul Fatt had collaborated with Bernard Katz for carrying out intracellular recordings at the NMJ. So that is one correct answer. So Bernard Katz and Paul Fatt collaborated to find out the to basically advance our understanding of the muscle end plate potential. What else? Any other option which is correct? So you have Charles Sherrington, Rita Levi Montalcini, and John Eccles. Ananya says the other option is D, John Eccles. Yes, Ananya, you are correct. B and D are the correct options. So with Bernard Katz, so Paul Fatt was basically an American whose contributions are often not like people often forget or it's not very well acknowledged. But Fatt collaborated with Bernard Katz, like I said, to study the neuromuscular junction and they did the intracellular recordings, like I said, using the frog neuromuscular junction. And with John Eccles, he studied the inhibitory synapses. So, like we now understand, there is not just excitatory synapses, there are also inhibitory synapses, right? So, inhibitory synapses are those where an inhibitory neurotransmitter is released. So, that will inhibit the postsynaptic neuron or muscle or whatever. So the postsynapse is basically inhibited. So there are both excitatory and inhibitory synapses, and John Eccles and Paul Fatt together studied the nature of inhibitory synapses. So yes, those B and D are the correct answers. Very good, Ananya. Does anyone know who Rita Levi Montalcini was? What was Levi Montalcini's contributions? Does anyone know? Again, this is something that you would have come across during the course lectures. What Rita Levi Montalcini had done, and for her work, what was she awarded? Does anyone know? Any guesses? Yes, Shrija, you are correct. Rita Levi Montalcini worked on nerve growth factor. For that, Rita Levi Montalcini, in fact, received the Nobel Prize. So, if you go back to the course videos and check out the part of the session on signaling molecules, so about the discovery of nerve growth factor, that is where you will find out about Rita Levi Montalcini. Again, go back and read about the struggle that Rita Levi Montalcini had to go through, because at that time World War was happening. Rita Levi Montalcini was an Italian, and there was a lot of problems happening in Italy. She had to flee, she had to stay in disguise under false names, and she really struggled in Italy at that time because of the World War. Do go back and check out all of these things. It'll be a very interesting read for you, and it'll also help you understand the kind of struggles that scientists had to undergo during times of war and times of like these kind of tumultuous times, and what all they had to endure, and how they actually persevered in the face of all those problems, and how they actually came up with these really groundbreaking discoveries, all because of their perseverance. So do go back and check that. So the correct answer to this question, so the two correct answers are Paul Fatt collaborated with both Bernard Katz and with John Eccles. Next question. Which of the following are Nobel laureates? Again, it's a multiple select question. So is it John Eccles, Julius Bernstein, Rita Levi Montalcini, or Stephen Kuffler? So which scientists among these are Nobel laureates? So Anushka says C. So C is, of course, we just discussed in the previous question that Rita Levi Montalcini is a Nobel laureate. Okay. Is that the only Nobel laureate? Do you know any other Nobel laureate in the list? So what about Bernstein? What about Eccles? What about Kuffler? Do you know if any of them are Nobel laureates? Of course, Eccles was also a Nobel laureate. So both Eccles and Montalcini were Nobel laureates. Of course, they did not receive the Nobel Prize together. They received the Nobel Prize during different times, but they were both Nobel laureates. We have discussed about the many contributions of Eccles. We have discussed about Rita Levi Montalcini's work on NGF. We discussed about Bernstein's membrane theory. Again, Stephen Kuffler, go back and check. Stephen Kuffler has also come up multiple times in the course lecture videos. So do go back and check about what he contributed and what kind of extremely elegant experiments Stephen Kuffler actually came up with. Stephen Kuffler was known for his extremely elegant experiments. Very, very simple, elegant, and actually explained the phenomenon very beautifully. So go back and check that. That's your homework. You will, of course, find it in the course lecture videos. Next question. Which of the following did Paul Fat work on? We just discussed this, so I'm quite sure you all will know the answer. It's a multiple select question, so there are multiple correct answers to this question. So A, muscle end plate potential, B, the nature of synaptic inhibition, C, action potential, and D, active properties of dendrites. Again, who's going to answer this question? Again, let me show the question to you. Muscle end plate potential, nature of synaptic inhibition, action potential, or active properties of dendrites. So, what did Paul Fat work on? We just discussed this. Okay, Shrija says B, the nature of synaptic inhibition. Okay, that is one correct answer. Very good, Shrija. What else? Is that the only answer? Is there any other answer that is right? Recall. We just discussed these things. We just discussed what Paul Fat did, whom he collaborated with, what kind of work did he do, how he was, like, although he made these contributions, how it was not very well known. I mean, his contributions to these things were not well known. Anushka says A. A is muscle and plate potential. Very good, Anushka. Both Shrija and Anushka, you are right. A and B are the correct answers to this question. So, like I said, for muscle and plate potential, he worked with Bernard Katz. And for this, he worked with John Eccles. Interestingly, do you know? Again, if you go back and check the course videos, you will get to know this that in fact, Katz and Eccles also collaborated with each other. So, although Paul Fat collaborated with Katz and Eccles also collaborated with each other. So, how did that happen? Let me give you a brief, let me tell you a brief story about all of this. So, Katz was basically, he was born in Leipzig in Germany. He was a son of a fur trader. So, fur. So, he was a son of a fur trader. And at that time, there was in the place where he was living, there was Russian hegemony. But then there was, after that, the Russian Revolution came. And because of that, he ended up becoming stateless. Okay, so he was educated in Leipzig, Germany. He did his MD degree at the University of Leipzig in Germany. And but at that time there was a lot of anti-Jewish sentiments. A lot of people were against Jews, and it was very getting very difficult for Katz to continue to stay over there. So initially he was thinking, let me just emigrate to Palestine and get away from all of this. But then he was convinced by other people that no, 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 don't do that. So then he had published some work which came to the notice of A.V. Hill. So A.V. Hill was a scientist in University College London, UCL. So, and he was a very famous scientist, and he was trying very strongly to oppose the Nazi, he was against what the Nazis were doing, and he was being very strongly vocal about that, and he was trying to help the war efforts in terms of that. So, Katz got a recommendation from one of his professors at in Germany, so that he could join A.V. Hill's lab. And then A.V. Hill also saw Katz's papers, the one or two works that Katz had published. So A.V. Hill was also happy to allow Katz to come to his lab at University College London and do a PhD. So Katz came to A.V. Hill's lab in around 1935, and he did his PhD over there. And while he was doing his PhD over there, Katz interacted with both John Eccles and with Hodgkin. So both he became good friends with both Eccles and with Hodgkin. So after his PhD, so like I said, when I told you about John Eccles, remember I said John Eccles was an Australian, and John Eccles was actually a student of Charles Sherrington at Oxford. So, but the thing was that John Eccles was very outspoken. He was very brash. He was not. He did not gel well with other people. Not as nicely. So yeah, he was not as well liked because of that. So after he finished his work, John Eccles could not stay in Oxford. So he had to leave because he was not offered a position to continue. So John Eccles thought that he might get a position, but he did not get. So he had to go back. So John Eccles went to New Zealand, and he went to Australia and so on. So when John Eccles was in Australia, he asked Katz, "Hey, why don't you come and work with me? We can collaborate on these things." So Katz went to Australia, to Sydney, and so the two of them collaborated. So I think around 2 years or so, Katz and Eccles were collaborating. Katz stayed in Australia to work with Eccles. Then after that, Katz worked in the military for some time. He served in the military because by that time, Katz had become a naturalized British citizen. So initially, like I said, he was stateless, but then he became a naturalized British citizen, and therefore he was able to serve in the war. So he served in the military, World War. So, during the World War II, he served in the military. I think as a naval officer or something. I mean, I forgot the exact designation, but he served in the military. And then after that, so I think for some around 4 years or something, he served in the military. And then he got married, and then A.V. Hill said, so as a wedding present, A.V. Hill said, you know what, why don't you come back, come to University College London, and I'll help you get a position to come back over here and do some work. So then Katz went back to University College London, and there he became a professor of biophysics. So this is the whole story about uh, how cards basically interact. So his relationship with his mentor A.V. Hill, his collaboration with John Eccles, the fact that he served in the military, and of course the level of discrimination that he faced. So cards, being a Jew, faced a lot of discrimination. And the papers that he was trying to publish. So A.V. Hill gave him some papers and said that why don't you work on this? Why don't we publish it? He worked on it and he submitted it to a journal, and the journal said sorry. Only if there's an Aryan co-author, we will publish it. He is Jewish, so we won't publish it. So he faced all of these kinds of discrimination. It was very difficult for him. And then after the war, when he was trying to set up his laboratory at University College London, because of the war, like the situation was very poor. It was very difficult for him to set up a laboratory. There was a shortage of everything. The city of London was heavily damaged. So it was quite difficult for Katz to actually carry out research. But still, he managed to work on the neuromuscular junction and come up with the quantal release of neurotransmitter hypothesis. We also worked with Eccles, and that also led to some fruitful results. He also, like I said, he Katz also had a good friendship with Hodgkin. Remember, so of the Hodgkin and Huxley fame. So, together with Hodgkin, Katz also came up with the sodium hypothesis, the fact that sodium ions are probably involved in the action potential. So, all of these things, this is how the scientists of that time basically collaborated with each other, had good relations with each other, helped each other during the time of need. A.B. Hill even allowed Katz and his wife to stay for 2 years at his own home before like they could get a house of their own. So that was the kind of relationship that scientists possessed with each other, the kind of friendship, the kind of collegiality that they had, the kind of the kind of they way they worked with each other for the sake of science. So this is something that we should also try to understand and try to adopt during our endeavors in science. So yeah, I just wanted to give you a little background on cards. I wanted to give you a little background on Eccles and all of these things. If you go back and look at the lecture videos, all of these things are covered in different lecture videos. And please go back and recap those. It will be very useful for your final exam. Okay. Yes, Ananya. A and B, you are also right. Next question. Who received the Nobel Prize for the discovery of the nerve growth factor? This is something that we have already discussed. Who can say the right answer? Ananya says B. Yes, Ananya, you are right. Rita Levi Montalcini did receive the Nobel Prize for the discovery of the nerve growth factor. She did not receive it alone. Both Rita Levi Montalcini and Stanley Cohen received it together. So Stanley Cohen was a biochemist who was the one who actually purified the nerve growth factor. So both Rita and Stanley Cohen received the Nobel Prize together. And these other individuals also played a very important role, although they did so in the sidelines. Please go back and look at all of these individuals. So Frank Lilly was an embryologist who was also a supervisor or a mentor for Victor Hamburger. And Rita Levi Montalcini actually worked in Victor Hamburger's laboratory. So again, go back and check the story of this. So how Victor Hamburger generously invited Rita Levi Montalcini to his lab because they were getting some contrasting results regarding their experiments, and how that actually, so what was the role of Giuseppe Levi? So Victor Hamburger, I was actually discussing his work with Giuseppe Levi, and Giuseppe Levi actually passed on that the work and so on to Rita Levi Montalcini, and that is how this whole thing happened. So do go back and check about all of these individuals. Although Rita was the one who won the Nobel Prize, these other people also played a part in that story, different aspects of that story, but all of these individuals were involved in one way or the other in the discovery of the nerve growth factor. Is that clear? Okay, Shrija. Uh, Next question. Which animal model did Bernard Katz use for his discovery of the miniature inflate potential? Now, this is a very easy question. We have probably discussed it 3 or 4 times today. Yes, Shrija, you are right. The answer is the frog neuromuscular junction. Chick embryos was actually used by these people. Frank Lilly and Victor Hamburger were working on chick embryos. Again, go back and check. Salamander was used by many other scientists in many other contexts, but not by Bernard Katz for this discovery. Again, go back and check who used salamander and why did they use it. What was the So why did, like, when, how did scientists know that which model was good or which model was ideal for which kind of studies, and why did they use that particular model for that kind of study? Do go back and check all of that as well. It will give you a lot of insight, because even now when scientists carry out experiments, can carry out studies in live organisms, the model of choice depends on a lot of factors. And one of the most important factors is the ease with which you can perform experiments with that model. How easy is it for you to not just grow that model, but also to visualize or get the kind of data that you want from that model. So, for example, when Hodgkin and Huxley used the squid giant axon, why did they use the giant axon of the squid? Because it was very large. It was very easy for them to insert their electrodes inside. In case of salamanders, in experiments where you wanted the nerve to regenerate, for example, and you know that in salamanders and newts and so on, the regeneration potential is very high. So for those kind of experiments, it made sense to use salamander. So in different cases, depending upon what your objective is, you will use the appropriate model organism. For example, Eric Kandel, do go back and check on about this, about Eric Kandel. He also again won the Nobel Prize. Why, do go back and check on why he won the Nobel Prize, what did he do, and so on. So Eric Kandel used aplasia too. So it was a, why did he use aplasia? What was aplasia? And how did he use aplasia to understand learning and memory? Do go back and check. So, is that clear? Any questions? No. Okay, none so far. Next question. What did Stanley Cohen use to purify the nerve growth factor? Was it Bucher's tumor? Was it snake venom? Was it chick embryo? Or was it mouse submaxillary gland? What was the system that he used? Anushka says snake venom. No, Anushka, that is not technically correct. Snake venom did play a part in that story, but it was not it was not what he actually eventually used to purify nerve growth factor. Snake venom was involved. Yes, you are right. It was involved, but that was not the that was not what he actually used to purify nerve growth factor. Any other answer other than snake venom? It's okay. Just guess. It's all right. We'll discuss. Any other answer? Anushka says D. Yes, Anushka, that is actually correct. Stanley Cohen did use the mouse submaxillary gland to purify the nerve growth factor. So, how did it go? Like I said, in Victor Hamburger's laboratory, Stanley Cohen was a biochemist who had come for his postdoctoral studies. So he already had a strong biochemistry background. All right. So they wanted to purify this protein, which they were seeing is responsible for this um, rapid growth of rapid nerve growth. So there was um, a friend of Victor Hamburger called Bucher, who had told him about the fact that in his if he had a tumor, he saw that in tumors derived from the nervous system, he was able to see that there was a lot of growth of nerves close to the site of angiogenesis. Okay, so using this, Victor Hamburger and Rita Levi Montalcini carried out further experiments using this tumor model. Okay, and then from that, they realized that there is probably some protein that is responsible for this growth, and they wanted to purify that. So that's when Stanley Cohen came to the picture. So they realized that snake venom probably has a lot of this nerve growth factor. They wanted to use snake venom in some other context, but while doing so, they realized that snake venom actually has a lot of the nerve of this protein, which is responsible for this nerve growth. So snake venom actually is produced by the submaxillary gland of the snake. So they thought, okay, in that case, instead of using the snake, let us check the submaxillary gland, but of the mouse. So Stanley Cohen used the mouse submaxillary gland to purify this protein, which they called as nerve growth factor. So actually snake venom they were using as a kind of a control for their experiments, but they saw that actually instead of acting as a control, there was a lot of the same protein in the snake venom. So that is the link between snake venom and the purification of nerve growth factor. But they it did not actually use snake venom to purify nerve growth factor. They actually used the submaxillary gland of the mouse, because they know that snake venom is also produced by the submaxillary gland of the snake. But it was, I think, maybe, I mean, I'm speculating here, maybe it was easier for them to access the submaxillary gland of the mouse. So that's why Stanley Cohen used the mouse submaxillary gland to purify the nerve growth factor. So I hope that is understood. So the correct answer is not snake venom. The correct answer is mouse submaxillary gland. Next question. True or false? Nerve growth factor has both neurotropic as well as neurotrophic actions. Can anyone answer? Is it true or is it false? Shrija says it is true. Yes, Shrija, you're absolutely correct. It is true. Nerve growth factor has both neurotropic as well as neurotrophic actions. Again, this is something that has probably been discussed in the first set of video lectures. So I'm not going to repeat what neurotropic and neurotrophic mean. It is I would strongly suggest that please go and check the, I think in the first week or the first or second week video lectures, you will find the definitions of neurotropic and neurotrophic. But Yes, this statement is true. Nerve growth factor is both neurotropic and neurotrophic in nature. And this is not just true for nerve growth factor. There are many other growth factors also which have both neurotropic and neurotrophic actions. So I leave it to you to check what neurotropic and neurotrophic mean. You will find these in the first in the I think in the first or second week video lectures. You will find the answer to this. What neurotropic and neurotrophic. I mean the meaning of neurotropic and neurotrophic. I think it's in the first weekly video lectures. Please go and check it out for yourself. Next question. Which of the following was proposed by Paul Weiss? Again, it's a multiple select question. So, multiple answers are correct. So, is it the resonance theory? Is it the chemoaffinity hypothesis? Is it the contact guidance theory? Or is it the law of dynamic polarization? So, who is going to tell me? Which of the following was proposed by Paul Weiss? We discussed this, right? We discussed it in one of the previous questions, right? What regarding when we're talking about Paul Weiss, we said that he was a neuroembryologist, and we discussed that he had come up with certain proposals. Anyone? Okay. In that case, let me give you the answer. So he had come up with the resonance theory and with the contact guidance theory. Okay. So Paul Weiss had come up with these two different theories, the resonance theory and the contact guidance theory. Whereas the chemoaffinity hypothesis was proposed by the one, Roger Sperry. So Roger Sperry was actually had actually done his PhD with Paul Weiss. So Paul Weiss had been Roger Sperry's PhD mentor, but Roger Sperry had later come up with the chemoaffinity hypothesis, which actually was which actually disproved or which was opposed to the contact guidance theory and the resonance theory that were proposed by Weiss. So Roger Sperry, who had actually done his PhD with Paul Weiss, had proposed the chemoaffinity hypothesis. So this was proposed by Roger Sperry. Roger Sperry had been a student of Paul Weiss, but this chemoaffinity hypothesis that had Roger Sperry proposed was had disproved or was opposed to the contact guidance theory that Paul Weiss had proposed and the resonance theory. So Roger Sperry had disproved his own supervisor's hypothesis. The law of dynamic polarization was proposed by none other than Santiago Ramon y Cajal. So, what was the law of dynamic polarization? It was very simple, at least in current. So, he basically said that there is a unidirectional transfer of information from dendrites to the cell body to the axons. So this he said was a unidirectional flow. It happened only in one direction. There was a polarized direction of information transfer in the neurons, is what Cajal had proposed. And that is what he called as the law of dynamic polarization. What was the contact guidance theory? So, contact guidance theory, which was proposed by Paul Weiss, was actually was a theory to describe how neurons probably interact with or make contacts with their target cell. So Paul Weiss said that it is all probably just mechanical contacts. They basically just mechanically, there are mechanical interactions or mechanical forces which are responsible for contacts between between neurons and their target cells. So that was the contact guidance theory that was proposed by Paul Weiss. But then his own student, Roger Sperry, who had done his PhD with Weiss, had later come up with the chemoaffinity hypothesis, in which Roger Sperry said that no, it is not some mechanical, it's not because of mechanical contact or interactions and so on. It is not because of functional activity. It is probably because of certain chemical markers or certain bioidentification tags or certain chemical gradients which are present in the neuron and in the target cell, which are responsible for the specific interactions between the neuron and its target cell. So that was the chemoaffinity hypothesis that was proposed by Sperry. So the correct answers to this question are resonance theory and contact guidance theory. So resonance theory was basically a theory that Paul Weiss had come up with, in which he was equating he was saying that there is probably the interactions probably exist because of resonance. So the way a musical instrument like a violin has resonance, so there is probably a resonance in the neuronal cells which is responsible for their interactions. So that was the resonance theory that Weiss had proposed. So both the resonance theory and the contact guidance theory turned out to be inaccurate, and the chemoaffinity hypothesis by Roger Sperry actually triumphed. So I hope that is clear. Next question. What did Roger Sperry win the Nobel Prize in the year 1981 for? So for like when what work did Roger Sperry do, which won him the Nobel Prize? So who can tell me the answer? Is it the chemoaffinity hypothesis? Was it action potential? Was it split brain experiments and hemispheric specialization? Or was it quantal release of neurotransmitters at the NMJ? So what did Roger Sperry win it for? Shrija says A. What is A? A is chemoaffinity hypothesis. No, Shrija, this is actually a trick question. So, although Roger Sperry did propose the chemoaffinity hypothesis, he did not win the Nobel Prize for the chemoaffinity hypothesis. He proposed it, but that is not why he won the Nobel Prize. Shrija says C. That is right. Wow. He did win the Nobel Prize for his work on split brain experiments and hemispheric specialization. So, if you go back and look at the lecture 11 videos where the professor discusses the work of Roger Sperry, you will see that Roger Sperry performed experiments on individuals in whom the corpus callosum, so there's a region of the brain called the corpus callosum, which actually acts, if you can say, as a bridge between the left and the right hemispheres. So the corpus callosum is responsible for communication between the two hemispheres. So in at that time, what used to happen was in patients who had severe intractable epilepsy, in certain cases, the surgeon used to basically sever or cut the corpus callosum in order to basically treat the epilepsy. But Roger Sperry's work showed that this when so the individuals, it appeared that the individuals were normal, initially, but Roger Sperry's work showed that in these individuals with a split brain, where basically the two hemispheres were not directly communicating with each other, it showed that each of the hemispheres actually has a specialization. So each of the hemispheres is dominant in terms of processing certain kinds of information. For example, the left hemisphere is responsible, or it has more ability, or is more dominant in processing speech, language, logical and decision making, and so on aspects, whereas the right brain is has a greater specialization in processing like the visual and the spatial information, or in terms of creative ability, and so on. So basically, his work showed that the two hemispheres have different specialization, and also these individuals who in whom the two hemispheres are not able to communicate, those individuals actually had a problem in coordination. So if they had to use both their hands to like to button up their shirt or something. So in that case, their coordination was not proper, because each of the hemispheres controlled a different hand. So the right hemisphere controlled the left hand, and the left hemisphere controlled the right hand. So there was a problem in their ability to coordinate. Or if the right hemisphere reached out for an object, the left hemisphere, sorry, the right hand reached out for an object, for example, the left hand used to resist. Why? Because the two hemispheres were not able to communicate with each other. So his work also showed, he also made some interesting observations regarding consciousness based on these split brain experiments. I again urge you, do go back and look at, check out what kind of work he did, and how that actually improved our understanding of how the different hemispheres work, and how they communicate with each other, and how you identify as a single individual or single self. So do go back and check that. Okay. Like I said, Roger Sperry did work on the chemoaffinity hypothesis, but he did not win the Nobel Prize for that. Action potential, we know that the model for action potential was given by Hodgkin and Huxley, and not by Roger Sperry. Quantum release of neurotransmitters at the NMJ, can anyone say who worked on that? So who worked on understanding, who proposed the quantum release of neurotransmitters at the NMJ? We discussed this in a little while ago, right? Anyone? Bernard Katz, right? Yes, Ananya, you are right. So this was by Katz, and action potential was Hodge. So, when I say action potential, when I say that Hodgkin and Huxley, I mean that they came up with a model to explain how action potential actually works. So, Hodgkin and Huxley, right? Next question, final question for the day. Which of the following was proposed by Emil Fischer? The resonance theory, lock and key hypothesis, theory of evolution, cell theory. It's a very, very simple question to end the session with. So, who is going to answer this? Is it the resonance theory? Is it the lock and key hypothesis? Is it the theory of evolution, or is it the cell theory? Any volunteers? Shrija says lock and key. Yes, Shrija, we can lock that answer as correct. So, Emil Fisher was the first to propose the lock and key hypothesis. So he proposed that an enzyme, he was working in terms of sugars. So he had proposed that an enzyme and a substrate, basically the enzyme has a particular shape, okay, which can be basically only be filled by the correct key or the correct substrate which has the correct shape. So he was the one who proposed the lock and key hypothesis. So with that, I come to the end of today's session. I hope you had a very enjoyable session today. I hope you also understood the different concepts and the contributions of different scientists. You're welcome, Shrija. I'm glad that you uh, were able to understand, and I mean, all of the students who answered. I'm very glad that you guys understood the concept and you attempted to answer. So, with this, I come to the end of today's session. Thank you very much for joining. Uh, Good luck with your preparation for the exams. And with that, I will stop the presentation. I'll stop the meeting. So, thank you very much, all. Have a good day.